And we're getting a thumbs up here. Thank you so much indeed, Brian. Okay, I'm going to set it off. I don't have any background information on you, but I think you can talk about yourself pretty well. Welcome to Carl Carruthers. Thank you so much for being here today.
Am I using this? Hello?
Yeah, is this on? And you can always use the
Okay. I guess I'll just use this. I can speak pretty loud anyways. So hi everybody, my name is Carl Carruthers. I'm very happy to be here today to talk about things that I've learned in the last probably 10 or 12 years from flying things in space. I'm originally from Florida. I'm originally from South Florida. I grew up here and I moved to Texas almost 10 years ago. And that was for when I went to Texas A&M for graduate school. Here's my Twitter handle if anybody's interested, and my email if anybody has any questions. So currently, I work at the Methodist Hospital Research Institute in the Texas Medical Center. I've been there for about 4.5 years. And I work on the structure and biochemistry of proteins that are called nuclear hormone receptors. I can talk to you in depth all about that if you really want to hear about it, but it's really not the subject of my talk today. Thank you. So what I'm going to talk about today is just the goals of what my microgravity projects were, what worked, what didn't work, and please ask me questions about my experiences. I don't know everything, I'm still learning, and so I may not know the answer to something. Please ask me, I'll get somebody who will know the answer, or I'll point you to somebody who may know the answer, but please ask questions. So my first project that I ever did was a program way back in the day that started, I believe, on STS-4, and it was a project called the Gas Can Project, or Getaway Special Project. And what this was literally a can, and you had an option of either 2.5 cubic feet or 5 cubic feet. And it flew in the shuttle payload bay, and they were self-contained. You had only one option, and that was usually on or off that the astronauts would do. And they were relatively cheap for their day. I believe it was about $5K for a 2.5 cubic feet can and a $10K for a 5 cubic foot can. All this may kind of sound familiar. It sounds kind of like the NanoRacks, what they're doing with the cube, fill a cube with whatever you want. And you could literally fill this cube with whatever you want as long as it wasn't trinkets and within certain safety requirements. It was a great program. It flew all the way up until STS-107. And unfortunately, after STS-107 was canceled due to they needed all the space in the payload bay to fly parts up to the space station. But it was an awesome program, and I was wandering around in Broward Community College down in South Florida, trying to decide what I wanted to do with my life. Sorry. Probably kind of wandering around in community college, deciding what I wanted to do with my life. I'd taken 1 or 2 classes here. I had become friends with Professor Rolando Brantley, who's here today, and he got me involved in this project. And our original plan was to for a 5 cubic foot can. And it was an incredibly ambitious project. We had a hyperspectral imager in it. We had some inorganic protein crystal growth. There was just a ton of stuff in this 5 cubic foot can. So about 7 months before STS-91, which was in 1998, June of 98, they called us and they said, hey, would you like to fly on STS-91? But it's going to be a smaller payload. Of course, we said yes, because the 5 cubic foot can, even though we didn't have a manifest date on it, if you get an opportunity to fly, fly. Don't ever turn it down, especially back in the shuttle days. So we took our most passive experiment that was on the 5 cubic foot can, which would be the easiest to do in the shortest amount of time. So that project was called GREAT-X. One of the fun parts about doing experiments in space is you get to come up with fun acronyms. And GREAT-X stood for Genotoxicology Radiation and Dosimetry Experiment. And that's a big fancy title for essentially just looking at the effects of cosmic radiation on in vivo, which means in living things, like seeds, or in vitro, which means in solution, like just DNA floating in solution. And we wanted to see the effects of cosmic rays on DNA, which was simple, easy, passive, path of least resistance. It did fly on STS-91 in June of 1998, and it had about a 10-day exposure in the payload bay. So this was my first ever payload that I had ever had the chance to participate in. I always loved space. I remember watching space launches from literally my backyard in Florida, and it was my opportunity to actually get involved. I had always liked science, but I was not going down the path of science. Let's just say I really didn't know what I wanted to do. And it was because very much of this project that I got involved in that gave me a career and what I do now. And I'll always be proud of this little project that we did 10, 12 years ago. More than 12 years ago. Now, geez. Time goes by quick. So this is Grade X, very unassuming. There's not much to it. There's a can, a lid, samples, and a big piece of foam. But to NASA, this is a lot. This is biological material, and that's all you got to say to NASA to get them all sorts of excited. So these little tubes down here are the in vitro and in vivo DNA. Literally, like, they were just seeds, and we had different species of DNA in solution, which were donated actually from a researcher at Texas A&M. We had a big piece of foam, which these little vials went into. This big piece of foam with the vials went into this box, and we sealed the lid. Triple containment, little vial, this, and then finally the gas can itself. This piece, everything on here had to have a pedigree, had to have a list of where it came from and how it was made. NASA loves paperwork, and they wanted every single bit of information we had about all of the things that we put in this box, including like the aluminum, where this aluminum came from, where it was made, how we made it, etc. The box had to be subjected to vibration tests. It also had to be able to stand certain G loads within all three dimensions. This was also what's called a pressure vessel, and it had to be undergo pressure testing. So what looks like just a bunch of samples in a box was a great learning opportunity. I got to learn the legendary 12 inches of paperwork required to fly something in space. And it was an awesome opportunity. It sounds like a lot of work, and it was a lot of work. But the ability to work with a group of students and then the ability to actually fly something in space, we were there for not only the development of this payload, but we were also there for the integration, which means literally bolting it to the side of the back of the space shuttle, to the launch, to the deintegration, to getting our payload back, something that flew in space. And that was nothing more cooler to me than that. Something that literally was in space and exposed to cosmic radiation. So what we did when we got the DNA back, we did a simple gel assay. We just ran it on a gel. It's a real simple assay that we do. And you can run it, compare it against our ground controls. And if the DNA breaks up a lot, it smears a lot. If it doesn't break up too much, it doesn't smear as much. And that's really all it was. We compared that to our ground controls. The seeds were given to some kids. I forget where. They went to a lot of places, and I don't remember where they all went. We never saw the kids again. No, not really. They grew the plants, which I believe was a Rapidopsis, which is just a little weed, mustard weed it's called. And they looked at the plants and looked for changes in its phenotype or physical attributes of the plant itself. Those seeds also went on to inspire a whole other group of kids that got to deal with space seeds, which was once again, anything from space when I was a kid was awesome. So what did I learn? There's a lot of paperwork involved with putting anything into space. But 90% of it is due to safety requirements and is absolutely important. And I completely understand why, and it was an excellent opportunity. This experiment was completely passive. Passive is good. Automated is good. The least you have to deal with the project, the better it's going to be. Let's just say the more reproducible it is, and the less, actually it's less paperwork. We learned about the importance of ground controls. Having something on the ground in order to compare it to something in space is very, very important. And anytime you do science, when you're going to learn, the first thing you're going to learn is you only have two friends in science, and that's negative controls and positive controls. And they're very important. And that was one of my first big scientific lessons. So the next thing I went on to was Microgravity Flights. And this was a program called the Reduced Gravity Student Flight Opportunity Program, which I think now is Microgravity University. It still goes on. So I went to Professor Brandley, And I said, hey, they got this really awesome opportunity. If you create a proposal and send it to NASA and they select you to fly on this KC-135, they'll fly your experiment. And he goes, oh yeah, that'd be a great opportunity. It's a great idea. And I don't think he really expected me to do anything with it after that. But I was incredibly motivated from the last experiment that I flew. And I put together an experiment pretty quickly, and we got selected. And I said, okay, now I have to actually build this thing, and I have to actually create a team. And before that, none of that existed. It was just an idea on paper. For those of you though, who aren't familiar with the KC-135 flights, it's an aircraft called the KC-135, just like this, and it goes up at a 45-degree angle, tips over to the other side, and goes down at a 45-degree angle. A roller coaster, essentially. At the very top of the roller coaster, you have about 20-25 seconds of microgravity. It's a lot of fun for some people. Some people, it's not so much fun. So it was a great opportunity to try to design an experiment just within the parameters of this type of flight. So there were this time our experiment was not passive, so we had all sorts of restrictions. We had power restrictions. We had to have a dead switch, which you had to have the ability to turn this thing off with one switch your entire project. There were size limitations. I believe it was only within the aircraft itself. You had, I believe 6 foot up to probably about another 5 or 6 feet. You had a volume restriction as far as what you could use. There was passenger safety. You're going to have people floating around in microgravity, and then suddenly the microgravity disappears and they plummet to the ground. So you have to have certain restrictions as far as fluid containment. You don't want stuff flying in people's eyes or anywhere else. And we had to have lots of what were called bumpers. Anything that had a sharp edge had to be padded and made soft, and you didn't want anybody hurting themselves. You had a time limit. They advertised usually about seconds when you just come out of the microgravity, when you just begin the parabola, and about 5 seconds after you come out of the parabola. And the total time is maybe about 20 seconds, with about maybe 2 minutes of a 2G pullout. So we also had to worry about getting sick. So any of the team members became incapacitated, they had to also, you had to either be able to do the entire experiment yourself or have somebody else help you, or you had to, it was definitely something that I don't think a lot of people had thought about before they flew. So this is a rough diagram of what the experiment was. It was a big, large cylinder, and it had, so it was called as if, because nobody believed we would fly, so anyways. It was some more big fun words called acoustical separation of immiscible fluids. Essentially, how do you separate oil and water in microgravity? In microgravity, if you shake up a thing of oil and water, like salad dressing, it's not going to separate like if you put it on this desk right here. You don't have a gravity vector to separate things due to its different densities. So I came up with the idea, how would you separate immiscible fluids like this in microgravity without a centrifuge? Centrifuges are complicated, and I was thinking, could there be a simpler way to do this? So we actually had different cylinders that had different mixtures of either oil or water. We had oil was yellow and the water was colored red. And we had 2 little speakers on the side. And we were thinking to try to separate the oil and water with sound waves. Liquid is about 1,000 times water, at least it's about 1,000 times denser than air. If you could actually create sound waves that would create these little areas of concentration, could maybe assist in nucleating the fluid and pulling it out of solution. So once we got to microgravity, this is essentially what we did. We turned on the speaker. We had a sound controller, which we could use different frequencies, and we were going to videotape it and determine possibly if there was a good frequency at separating the oil and water. This is the experiment itself. Here's our container. We had a light here. There's a video camera here. The frequency generator is up here. And as you can see over here, it's a fairly good sized cylinder, which once again was a pressure vessel that had to be tested. All of this stuff that you see here had to be tested just like anything that we flew on the shuttle. It had the same limitations, same restrictions that on a shuttle payload. But what you can see here possibly in this picture, besides the fact that I have a little less hair now, there's a lot of people in the background having fun. They're playing around in microgravity, while Ken and I were working feverishly away. So this is one thing that we had not thought about when we originally designed the experiment. It was pretty labor intensive. within that 20 seconds, we had to grab one of the cylinders that was in this little case here, shake it up, put it back into this containment container here, plug it in, select frequencies, and record it. 20 seconds. We'd practiced, but being 20 seconds in microgravity is much different than 20 seconds in 1G. It took several parabolas for us to get a rhythm down, which we eventually got going. We got it, but we worked all 35 parabolas, the entire parabolas, and when we got done, we were tired. Luckily, neither one of us got sick. There were several teams, actually several teams with all the members were completely incapacitated. They were sick, and their experiments they didn't get any data at all, which is kind of sad. So this is what we learned. Shaking things in microgravity is not as easy as shaking things in 1 gravity. It's a lot more challenging to shake things in microgravity. Writing notes, I had the crazy idea of just writing things down as we did them. Writing in microgravity is not as easy as you may think, even though you try to hold yourself down. It's a much different, disorientating experience. Leaning over the experiment itself was not fun. We had designed this thing so it was flat on the ground, and we're constantly leaning over. Even though you're in microgravity, it's just, you tend to twist in just a single finger push and you go flying in another direction. Experimentally, I don't think it really worked. I never really convinced myself that it worked. And I think that's because the volume of fluid was just large, and/or we just didn't have enough power source, sound source to actually separate the oil and water. And once again, those guys that were having fun, all the people who were in the background having fun, that's because they had automated experiments. They literally had 1 or 2 buttons to push, and then they got to play. And that stuck in my head, believe me. It's like, I'm going to make the next time, if we ever fly, I'm going to make it as automated as possible. So this is as if 2. We actually resubmitted and flew again. And so there was, each flight was 4 members. And including me, all 6 other members went on to have science careers after this, which I'm proud to say, because they were once again community college students, and it inspired them to pursue a career in science. So my next project, as if 2, is a little bit simpler. We just had some needles, and at different heights. We had one that injected oil, and we had one that injected fluid. And the idea was that these fluids would come out, they would mix, and then we would try to separate them with sound waves, sound from a speaker down below. This is as if 2. As if you notice one thing, it's actually now a 2-stage experiment where we're not leaning over, but we're actually just kneeling. This is the same box, same camera. We have somewhat automated where I just had to push the fluid out of the needles, and we would record and it did its thing. That's all I had to do. It was pretty simple. Down here is a speaker. If you can see over here, there's a vacuum tube. We had a vacuum that once we were coming out of the parabola, we would actually turn on the vacuum and it would suck the water out of the fluid instead of it building up inside the container. This is a whole lot of fun. This is out of Hangar 990 at the Johnson Space Center. And this is the review board that goes over your experiment board before you fly. No pressure at all. These are the guys that fly the plane. These are the guys that are going to be in the crew compartment with you. I mean, it's no pressure at all. It was pretty intense. So what did I learn from this experiment? It's once again, there's so many things that you don't think about until you actually do these experiments, and it's a cool learning experience. And I tell so many people this: when you fail, if you're going into grad school or you're going into science, get used to failing. I mean, it's going to happen, and you learn a lot from failing. If you're doing it right, you will learn a lot from your failures. So get used to it. It's not a bad thing. Better ergonomics was a much better experience. We got to have a little bit more fun in microgravity. Fluids behave very differently in microgravity, and that was one of the main things that I learned from this. And I got a lot of experience now dealing with fluids in microgravity. It's great because I watch those videos by Don Pettit, and I know that. It's a lot of cool. I actually got to meet him recently and talk a little bit about fluid stuff, and he's a cool guy. But they behave very, very differently. All that gravity, things that happen in gravity are completely gone. And they behave more towards their tendency of what the fluid likes. If you have hydrophobic fluid, they tend to stick to hydrophobic things. If they're more of an aqueous or polar solution, they stick to polar things. So what would happen as the fluid would actually come out of the needle, the oil would actually come out and then reabsorb back onto the needle itself. So we tried different, I literally had to wham the plunger on the syringe to actually get the fluid to come out. And sometimes if you did it too hard, it'd fly across the experiment. But that was one thing we didn't anticipate. I'd never worked with fluids in microgravity. We're so accustomed to dealing with things in 1G, we just never would think of that. But now, that was one of the main things we learned is we had got some valuable data on how fluids behave in microgravity. Semi-automated process, again, if you can make it automated, that's the best. Especially nowadays, it's so easy to make things automated. Moving forward, after all this, I eventually went to graduate school at Texas A&M University. I graduated there a while ago. I'm now, like I said, in Houston. And I got the opportunity, once again, through Professor Branley, to fly 2 experiments on STS-134 and 135. I now work in the field of protein crystallography, so I had an awesome opportunity to actually try and grow some crystals on the space shuttle. 134 and 135 flew for 16 and 13 days, respectively. And we used an apparatus called the MDA, or Materials Dispersion Apparatus, that was leased through NanoRacks. So how do you make crystals? I'll make you all protein crystallographers in 10 seconds. Anybody ever make rock candy? It's a very similar process to making rock candy. If you take sugar and pour it in water, you'll reach a certain limit where that water will not soak up any more sugar. But if you heat the water, you can add much more sugar than you would if the water was at room temperature. So this is called a hypersaturated solution. And as you pour sugar into hot water, you have this high amount of, much more sugar in the solution than you normally would. If you put in a string or a stick into the solution and let it cool, the sugar molecules will actually nucleate onto the string or the stick and actually start forming crystals. And then you eat it, which is good. This is very similar to the process of making protein crystals. We usually high concentrations. We try to remove as much water as we can, and they form little crystals. And these crystals aren't like salt crystals. They're actually more like Jell-O. They're between 40 to 80% water, so they're pretty squishy. And why do we grow crystals? So we grow crystals to determine the 3-dimensional structure of proteins. We take these crystals and then put them in front of high energy X-rays. The X-rays hit the proteins and scatter. From the pattern, from the scattering, we can then determine the 3-dimensional structure. And it's really, really important for drugs, drug development, and basically just how proteins work. To see things at an atomic level, you get a much more deeper understanding of how something works. It's kind of like always describing to somebody what a car is, which they've never seen a car. And then you finally give them a picture of a car. And they go, oh, okay, so you can kind of see where the person sits and how you may interact with the car, et cetera, et cetera. The resolution is also very important. If I give you a picture of a fuzzy car compared to a picture of a high-definition car, it's much better understanding. So the 3-dimensional structure of proteins is a very important field. This is a really quick diagram of how the MDA works. It's a really simple device. It consisted of 2 blocks, and then within the 2 blocks had little holes. Upon reaching orbit, an astronaut, which is another cool thing, I actually had to get to have astronauts at my bidding. They actually turned a knob. Not much, but getting astronauts to do things for you is pretty cool. They turn a knob, the blocks slide together, and the 2 solutions, whatever, are in the tubes get to mix. Then just before landing, they turn the knob again, and the blocks slide back into position. And it's within here that the protein crystals grow. I think this company also used this MDA for all sorts of various different experiments as well. So dealing with this experiment, I didn't design it. So there's a lot of things I didn't get any input into, which is for scientists is not fun. Scientists tend to want to control things, and to actually hand my stuff over to somebody else and let them do it is not. You're always worried, especially if something doesn't work. You're always worried at least when you've done something 100%, you've touched something or worked with something, you possibly know where things could have gone wrong, but when you hand it over to somebody else, you have to give them a lot of trust, and that was something I had to deal with. The volume that we used in these blocks were huge. They're about 100 times larger, more than 100 times larger than I'm used to dealing with, so I had to recalculate completely for how these proteins would actually crystallize in a larger volume. There was no control over the individual experiments. Once they were put into these blocks, you were in that block system with a bunch of other experiments. So it was all, you were treated like everybody else. And so there was another lack of control that I didn't enjoy, but it's, you know, hey, I'm gonna fly in the space shuttle, I'm not gonna complain. Ambient temperatures was another worry to me. Crystals tend to not like changes in temperature. They can form crystals, they're very temperature dependent, so you can literally go from one temperature where the crystal's happy, change the temperature, and the crystal completely goes back into solution. So we had no control over temperature at all, and that kind of worried me in the beginning. And there was a possibility of launch delays. So I had to make up multiple backup samples, and we also had to worry about ground controls syncing, just in case of the launch. It was something I hadn't had to think about before, and it was interesting to think about. This is the MDA being flown around by the crew of STS-134. Once again, you get to get your astronauts to do your bidding. It's fun. This is the actual MDA. That's the knob that they turned, and then they just kind of put it back in the mid-deck locker and let it sit. Nothing very complicated. Once again, easier is better. So sadly, when I got from both STS-134 and 135, I got no crystals. Sadly, that's the bad news. The good news is I get to fly twice in 6 months, and that's unheard of. And it's mostly thanks to the NanoRacks guys. Literally, my favorite story with NanoRacks, Mike himself and an intern rented, because 135 was a circus. I mean, it was everybody and anybody at the 135 launch. And getting in and out of the Cape is, if you're going to go anywhere from the Cape, it's like an hour to get out and to get in and to get out. So these guys rented an RV and lived in the parking lot of the Space Life Sciences building for 3 or 4 days. And it was awesome. That was my first interaction with NanoRacks, and I'm like, these guys are awesome. They're willing to, and this was in July, and it was with the heat, and they were really pretty looking at the end of about 3 or 4 days. They were, remember he was talking earlier about living in a small quarter for a very long time? Yeah, they were ready to leave. But it was awesome that they did that so they didn't have to waste time leaving the Cape and coming back all the time. They actually stayed in the parking lot, and that was I hadn't seen that kind of dedication to a flight before. So we determined in the end that it was possibly the apparatus. The apparatus had the two blocks, but they had a layer of grease in the middle, and I didn't know anything about that until I actually saw them like mayonnaise putting grease between the two blocks, and it was for lubrication for when they actually slid the blocks together. So it's possible when they slid the blocks together that stuff globbed up and got actually in the holes. I don't know. Or secondly, it could have been just due to temperature. There's so many possibilities for temperature changes when upon putting your proteins in the solution, and by the time you actually get it into the vehicle, by the time the vehicle launches, comes back, there's so many temperature fluctuations. More than likely, that's what happened. The crystals probably may have formed and just melted. I'll never know. So just some final words about my experiences. The ground controls and even an internal control, if you can make it specifically for a bio experiment, is really, really important. Think about that hard when you're designing an experiment. Sometimes your experiments don't work, like I said. You're going to fail, and it's science. Get used to it and learn something from it. But as Mike said earlier, their motto is, you can cheaply fail. At least this is different than before. Previously, when you flew something on station, if you had something fail, you were lucky to fly again within 2 years, if not more. You were lucky to fly if you flew. You were lucky to fly again, shall I say. But now I think it's really great that the process is getting what I say. It's moving at the speed of science, not at the speed of paperwork. And like I said, even if it fails, it's really, really important just to figure out why it didn't work. Learn something from it, and if you fly again, design it for those changes. Before you fly, test, and test again. Retest your experiment over and over and over again. Because it's going to be literally out of your hands and 260 miles up, moving at 17,000 miles an hour, over 17,000 miles an hour. Make sure it works. Try to test it just as much as you can. Have a backup plan. If not to get your your initial data, try to design try to design your experiment so you have some ability to get a secondary data set from something. Just if you can't get your primary data set, try to get at least a secondary data set. Automation, please, like I said, it's easier now than ever to make something automated. Use it. It's a gift. It's a gift that we didn't have in the old days. Make it as simple as you can, and most importantly, just have fun with this. I mean, it's a team building experience, and just have fun with it. It's a great opportunity, and I wish you all luck in this process. And that's all I have. That's really all I have.
Great. Really good. We probably have a couple of parking fines for you over in the SL.